Morning, everyone. Thank you so much, Jen. I'm actually here in my role as a scientific advisor to the Healy Center this morning, and I'm so excited to share this year's Student Lalji Award winner. Just a few words about this award. So thank you for allowing us to come here and share this with everyone. The 2024 Dr. Ayeez and Shelena Lalji Family Student Award is really special to us, and especially this year because we have lost Dr. Ayeez to ALS. It's been a few months, and it's really fresh for all of us and very sad. So in his honor, I'm just really happy to be here to do this. Just to remind them, I apologize if you hear a noise. It's my cat who's being very loud this morning. So please bear with me, and I apologize for that. But basically, the student award, the Scholar Student Award, recognizes and attracts the brightest young scientists into the field of ALS and facilitates meaningful research to understand the mechanisms underlying axonal and neuronal repair and regeneration in ALS and other neurodegenerative disorders that may be translatable to ALS. The selection is based on the academic credentials of the student, the scientific merit of the proposed project, and the relevancy of the proposal to axonal and neuronal repair in ALS. We are grateful to the Lalji family for giving this opportunity to the Healy Center to recognize these really brilliant students doing amazing work. We had a lot of applicants, and without further ado, we will announce this year's winner. And I don't know why my slide, there we go. Sorry, there we go. So this year's award goes to Anushka Sanyal, who's an undergraduate student studying human biology at Stanford University, under the mentorship of Dr. Erin Gittler, and her project is a novel RNA-based chaperone that protects against TDP-43 toxicity in ALS. Congratulations, Anushka. We can't wait to hear your presentations. I'm gonna now pass it to you and stop sharing, and you can take it away. Thank you. Congratulations.
Well, thank you so much, Dr. Sathru-Vikili. And really, before I begin my presentation, as I share my slides, I want to express my gratitude to both the Lalji family for their generosity, as well as the Healy and AMG Center for ALS for their research to support individuals with ALS and, their recognition through this award. So to start with this work, I am really excited to share more about elucidating the protective role of intronic RNA lariats against TDP-43 toxicity in ALS. So to get started here with a little bit of background, ALS is characterized by the aggregation of ubiquitinated proteins in affected motor neurons, and chief among these is TDP-43. And normally, TDP-43 functions broadly in transcriptional regulation, RNA splicing, and metabolism. But in disease, there is both a loss of function of TDP-43 in the nucleus and this toxic gain of function in the cytoplasm, where these aggregates sequester other essential RNAs and RNA binding proteins. So really, this motivates us to better understand how we might be able to target TDP-43 and through this really develop strategies to target it. Now, switching gears a bit, the DBR1 enzyme is responsible for cleaving the branch point site of looped intronic RNA lariats that are generated through splicing. And the deletion of DBR1 has been shown to increase steady-state levels of this lariat RNA. And now this is relevant because previous work done from my lab has established that the deletion of DBR1 acts as a suppressor against TDP-43 toxicity. And as you can see on the right, increases cell viability in mammalian cell lines and primary rodent neurons. So based on this, we sought to demonstrate how this deletion of DBR1 may ameliorate TDP-43 toxicity in yeast. As you can see, wild-type cells expressing TDP-43 deteriorate at a significantly faster rate compared to those cells that express TDP43, but also contain that DBR1 deletion. Looking to the right, we see that by fluorescently tagging these TDP43 aggregates and visualizing them using confocal microscopy, we find that cells that possess that DBR1 deletion seem to have fewer but larger, more spherical aggregates, which thus seem to be more dynamically structured. So this really brings us to the following model. So under conditions of moderate cell stress, it seems like aggregates are typically transient and can disassemble once a stress is resolved. Oftentimes, chaperones are recruited to facilitate this. But if the stress is prolonged or protective mechanisms fail, these can become permanent, forming large, insoluble, toxic aggregates. So perhaps while the TDP43 overexpression induces this assembly of static insoluble aggregates, these may be modified by the chaperone-like effects of this DBR1 deletion. So to better understand this, we really need to answer a few additional questions. First, it is unknown whether the deletion of DBR1 may confer a protective role in other forms of proteotoxic stress known to induce protein aggregation. So by evaluating this, we may be able to reveal more about the role of intronic RNA lariates in how these interact with TDP43 aggregates. Second, it has not yet been evaluated whether intronic lariat RNA directly can regulate this aggregation-related toxicity. And so in order to identify this, we can use a few follow-up experiments to really characterize the role of lariat RNA in modulating TDP43 toxicity. And finally, we would need to determine whether lariat RNA accumulation operates independently from classical chaperone systems to offer an alternative pathway to protect against neurodegenerative disease-related protein aggregation. So I'll quickly highlight a few key findings which we have used to start addressing some of these questions. So to evaluate proteotoxic stress and protein aggregation, we use acute and chronic stress assays in which we can see here that both in this survival assay as well as this spotting assay, the cells with the DBR1 deletion display greater tolerance against these forms of stress, really establishing the DBR1 deletion as a general protectant against protein aggregation and the toxicity that ensues. Next, we introduce an in vivo reporter that allows cells to translate lariat RNA species and accumulate these in wild-type cells. So while the wild-type BRA cells contain a wild-type reporter, the wild-type BRG cells sustain a point mutation at the branch site, which inhibits its degradation by the DBR1 enzyme. And so therefore, it can induce cytoplasmic accumulation of lariat RNA. Now, compared to the wild-type BRA cells, the wild-type BRG cells seem to better withstand this sodium arsenite treatment, suggesting that accumulation of lariat RNAs is sufficient to confer a level of protection against this stress-related protein aggregation. And finally, the inhibition of HSP104 can allow us to test the inhibition of one of the most potent chaperone systems implicated in responding to proteotoxic stress in yeast, and its activity can be inhibited by treatment with guanidine hydrochloride. So upon this treatment, we see that wild-type cells show both a decrease in survival as well as growth, whereas the DBR1 deletion cells remain unaffected. And so we can see that DBR1 deletion and the ensuing lariat RNA accumulation may offer a mechanism that operates independently of this HSP104 chaperone system to promote proteostasis. So these are a few summary findings, but generally, based on these findings, we are continuing our work to better characterize how intronic RNA lariats may serve as a way to buffer or chaperone against various forms of neurodegenerative disease-related aggregation. So before I end, I really want to thank the Lalji family for their support once again of this work and of young scientists as they instituted this award, and as well to the Healy and AMG Center for ALS for all of their work again, advancing ALS research. And lastly, I'd like to thank ALS ONE for this platform to share my work, and to all of you for listening to my presentation.
That's amazing. I mean, incredible work. And what it's just an honor to see you sort of getting started, because soon you'll be the keynote speaker at conferences like this with this kind of work. Honestly, that's just remarkable. And what a way to kick off our day.